Okay, yeah, so my name is Jakub, and I used to work at NBIS with Åsa and all the others, and I was together with some colleagues responsible for a course in epigenomics data analysis. So these slides and this talk come from that course actually, but I've been also giving it on the single cell course. So let's see now. Yeah. Okay, so what we're going to talk about today is what is epigenomics, and especially what ATAC-seq is, and also a bunch of these epigenomics experimental methods, how they've been applied to single cell, and a bit about multi-omics, where you combine data from several different assays, for example, measuring proteins and RNAs at the same time. And also a bit about spatial methods. And the focus is going to be on these two first things. So what is epigenomics, and especially about the single cell ATAC-seq data, and also how you analyze it. And I mean, this probably it's going to be less than an hour. We have an hour in the schedule, but I think this is going to be quicker, so you can go and enjoy the sun afterwards. And it's a very sort of broad overview. It's not much details, but it's yeah, mostly focused on general understanding. Okay, so epigenomics is yeah, starting with the epigenome. It's a bit of a contested term, but one common definition is that the epigenome is all the chemical compounds and proteins and chemical modifications that happen to the DNA that are not part of the the nucleotide sequence, but things attaching to the DNA in various ways, which can regulate gene expression. So they can turn genes on and off, and thereby controlling which proteins are made in which cells. And yeah, this is important for example, development and cell differentiation, and in cancer and in many other areas. And the field where you study this is called epigenomics. So basically, this is where people try to measure and analyze different molecules that attach to DNA across the whole genome, and to understand this. And yeah, you can see this. This is a common. There are many images like this, where you see the DNA, which is then wrapped around these nucleosomes, which are protein complexes of histones, and then the DNA is further coiled into these fibers and to larger and larger substructures until you come up with a chromosome like that. Okay, so yeah, as I just said, the epigenome is involved in many different processes, like development. Also, when it goes wrong, it can lead to cancer. It's implicated in aging and many diseases. And the things people usually measure when they measure the epigenome is DNA methylation. So when methyl groups are added to DNA, chromatin accessibility. So this is which DNA is accessible to other proteins. Basically, which DNA is not wrapped around, for example, these histones, or which DNA is not in some super condensed regions. Also, DNA protein interactions. So if you have a transcription factor binding to DNA, you want to see where this is binding, or if you have a histone with a specific histone modification, where are those histones located across the genome, or other proteins. It can be, replication complexes, things involved in DNA damage response or whatever. And many of these methods were first developed for bulk samples and have then been adapted to single cell. And ATAC-seq is probably the most commonly used one. So ATAC stands for Assay for Transposase Accessible Chromatin using sequencing. So the idea here is then to measure which chromatin is accessible to other proteins. Basically, what you do is you have an assay where you just sequence the part of the genome that's accessible, that's not wrapped around the nucleosomes or are in these condensed structures like heterochromatin. And the way this works is that you have this TN5 transposase, and you transfect the cells with this transposase, and it inserts itself into the genome. And this transposase has been a bit modified, so it actually inserts sequencing adapters to the genome. And some other tags that you can keep track of. And then based on this, you can, with PCR, you can purify this DNA that has this transposase inserted on either side of it. If it has it on both sides, you can sort of select for this DNA and purify it and sequence it. And that way you will get only DNA where you had transposase on both sides, which is in these accessible regions. And this works well. It's a quite easy protocol, and it works with very little starting material, including even one cell, which is what you want here. So accessible chromatin, also called open chromatin, it tells us, for example, where promoter regions are, if these promoter regions are active, or if they are sort of so if a promoter region is open, it means either it's active or it can become active at any moment. If a promoter region is full of nucleosomes, it means that the expression of this gene won't be turned on. You can also look at accessible chromatin to find enhancers, to find what this chromatin looks like around specific transcription factor binding sites. You can see how heterochromatin spreads across the chromosomes and other things. And this is often used as a complement to RNA-seq. For example, you might want to, you have your cell types called from RNA-seq, which I guess you talked about, and then you can use ATAC-seq to further refine those cell types into sort of subpopulations. Yes. The data looks like this. So basically, you have these DNA fragments that you have sequenced, and they are of different length. So some are long and some are short. And the ones that are shorter than the length of a nucleosome, so I don't remember how much it is now, 150 something base pairs. The ones that are longer than 150 base pairs, that you can assume that these transposition sites happened on either end of a nucleosome. That is this peak. And that's not what you're interested in. You're interested in these really short fragments. They are the places where these transposition events happen close together. So this is the actual open chromatin. So you get a bunch of these short fragments, which is what you want to look at. And then you look for peaks. So these are just places in the genome where you have lots of reads mapping from these short fragments where they just stack up. And there are many different programs for finding those peaks. One is called MAX3, but there are many. This is like a very old area in bioinformatics. Many years ago, people wrote a lot of these programs, and there's a lot of them out there. But yeah, this MAX3 is one of the most commonly used. So what this little cartoon shows is that these balls here are nucleosomes. So this is this DNA wrapped around these histones. These things here, I hope you can see my pointer, are the insertion events, and then you get different reads from this. So if you get a read just spanning this region, these reads are typically quite short, and they stack on top of each other and form a peak. If you have a read spanning from here to here, these reads are longer and correspond to these reads, and they are not what you're interested in. Yeah, so this is basically how ATAC-seq works. And this was very easy to develop into a single cell method. It was made by the same group that invented ATAC-seq, the Greenleaf Lab. And I think the single cell paper came the same year as the bulk method paper came. So they are almost like single cell ATAC-seq and bulk ATAC-seq are almost equally old. And now this is also available as a kit from 10x Genomics. So what you do here is you have your cells, and you add this transposase to all these cells together in bulk, and then you run them on the 10x machine. So each cell ends up hopefully in its own droplet, and then you get these DNA fragments which have the transposase, which is the black sequence, the sequence inserted by the transposase on each side, and then you get these cell-specific barcodes. And then you just sequence them all together. So this is, in a way, it's very similar to what happens in Single Cell RNA-seq. There is also an alternative method, which is called combinatorial indexing, the Single Cell Combinatorial Indexing ATAC-seq. So this is sort of what Parse Bioscience do for RNA-seq, and you can also adapt that to ATAC-seq. So I don't know if you have gone through this already in this course, these methods, Åsa.
Yeah, Henrik explained about the split pool barcoding for
Yeah, okay.
cell.
So I can so I don't have to go over this again. I'll just say that you can do this with ATAC-seq. As far as I know, there are no commercial kits available for this, but if you're big and ambitious lab and have some resources, you can make this work. And the good thing is that, if you have a big project, this becomes very cheap per cell, so you get a lot of data out. Of course, you lose a lot of cells along the way, so you shouldn't do this with some very precious, limited starting material. And this is what the single cell ATAC-seq data look like. So here's part of the genome. On the x-axis is just the coordinates. On the y-axis is how many reads you have stacking up. And this is how many reads you get if you combine a lot of cells together. You just, pool the data from many cells and look at them together. And then you can see clear peaks where you get a lot of reads. So this is where you have this open chromatin. And this top plot in blue, that's if you look at data from 4,500 cells pulled together. The bottom here is if you have 280 cells. So you see it still looks quite good. You can still see these regions of open chromatin. And then here you have data for the individual cells. And here you can see that if you look at an individual cell, the data is very sparse and very noisy. So if you look at an individual cell, it's just one read here, one read here. They're sort of scattered, and it's very hard to say anything conclusive about a single region in a single cell. You might have a read, and that should indicate that probably there's open chromatin. But if you don't have a read, you don't really know. Maybe it's just a dropout. Because the difference between single cell RNA-seq and ATAC-seq is that in RNA, you have many, many copies of the same mRNA in every cell. But here you just have, 2 copies of a chromosome. So at most you can have 2 reads from the same region. And usually, since this is not super efficient, usually you have 1 or 0 reads for each place in the genome. Yep. So if you combine, here you saw that if you combine data from many different cells, you get something that makes sense. You can also, for each individual cell, combine data from many different regions. And then you also see interesting patterns that make sense. For example, here you just combine data from all annotated transcription start sites, and then you see a clear peak that, like on average, there's a much higher signal at the transcription start site than around it. You can also look at specific transcription factor binding sites, so DNA sequence motifs that represent known transcription factor binding sites. And for example, here in this study, they looked at all instances in the genome of this motif. And then if they pooled for just one cell, the ATAC-seq signal, then they could see very clear patterns. So if you just look at one cell in one region, it's basically crap. But if you combine many cells, or if you combine many regions, then you get useful signals. Okay, so this is just a rough outline of how you usually analyze this data. When I was teaching this in this epigenomics data analysis course, then we also had the lab to go along with this, and you can probably find it online if you sort of are interested in looking more in detail of what this is done. But yeah, many of these steps are the same as for single cell RNA-seq, but then there are some peak calling, peak annotation, for example, are specific to ATAC-seq. Okay, so this first step is just demultiplexing, finding out for each read which cell it came from, clean up the reads to remove primer sequences, map them to the genome, use a different mapping method. Here you use BWA instead of star that you typically use for RNA-seq. But otherwise it's pretty similar. You remove duplicates because it's very, very unlikely by chance that you would get several reads mapping to exactly the same place from a single cell. So these are probably PCR artifacts. And you can also filter out some bad cells already at this stage. Then you do peak calling. So this is basically finding these regions in the genome where you have many, many reads stacking up. And you do this on aggregated data from all cells together. And you might also later want to go back and redo this for a specific cell type. Because maybe if you have a rare cell type with just a few cells, like 50 or 100 cells, maybe the peaks that are specific to this cell type will be missed when you do peak calling on everything together. But then what you end up with in the end is a count table like this. So this is also very similar to what you have for RNA-seq. But now instead of genes as rows, then you have peaks or genomic regions. This is maybe, chromosome 2 position, this and that. And then each column is then a cell. And usually this matrix is mostly zeros and it has some ones here and there. But yeah, the format is very similar to what you have in RNA-seq. Then, of course, you need to do a lot of QC on your data to see what has worked, what hasn't worked, which cells you need to remove. Here are some things that can go wrong in your experiment. You can have no cells in a droplet or many cells in a droplet, or maybe your cells are dead and broken. Maybe you have too few reads, so you can't say anything. Maybe transfection didn't work for a particular cell. For each cell, you collect lots of QC measures, statistics, and you use them with cutoffs to remove the cells you don't trust. So one thing you look at is how many fragments, basically how many reads you have in your peaks. If you have too few reads, you can't really do anything with the data. If you have a lot of peaks, maybe this is not a single cell, but several cells or clumps or something else that has gone on. You also want a substantial fraction of your reads mapping to the peaks and not somewhere else in the genome. So if you have very, very few of your reads mapping to the peaks, typically lower than 15 or 20%, then this tells you that maybe something has not worked here. And those cells you should probably remove. Yeah, you have these fragment sizes that we looked at before. So typically you have one peak. So if you make a histogram of all the fragments and their sizes, you have sort of a lot of very short fragments, and then maybe a bit of fewer fragments that span one nucleosome. So they're around 150 nucleotides. And then sometimes you even have some fragments that span two nucleosomes, so they're around 300 nucleotides. But if you have too few of these very, very short DNA fragments, that's also a warning sign. And then there's something called blacklist regions. So these are regions in the genome that have been defined empirically based on previous projects, for example, by the ENCODE consortium. So genomes, regions of the genome where this just doesn't work. You get a strong signal no matter what you do. No matter what kind of ATAC or chip or what experiment you do, you always get a lot of reads coming from these regions. Most of them are transposons or other repeats, but it can be other things too. So these are just regions that people have found that, we don't trust whatever signal we get from here. And if you get many, many reads mapping to these blacklist regions, then you should also, that's a warning sign. And then you also expect to see this, so an enrichment of reads around transcription, TSS, transcription start sites. Yes, so we basically calculate all this for different for each cell, and then you have some thresholds on which cells you want to keep and which you want to remove. Yes, normalization works very similar to what you do for RNA-seq data, single cell RNA-seq data. You basically account for sequencing depth. I think you do it slightly differently for ATAC-seq because you put different weights on different peaks. I think rare peaks are counted more. People have found that this works better. But yeah, it's to account for sequencing depth too. And then you typically project this to PCA, or here, singular value decomposition, which is a very similar thing to PCA.
Sorry, Jakub, I had a
Yes.
question. So what do you mean by more rare peaks will be valued more? Like peaks found in less cells?
Yes.
Okay, but still over some sort of a threshold, I'm assuming.
Yes. So first you do peak calling, and
Yeah.
then you need to get up to a certain threshold for this peak to be called at all. If
Yeah,
it's just
exactly.
like one read, then it's just noise. But
Yeah,
among the
exactly.
peaks that you identify, then peaks that are found in fewer cells are scored higher. They have more influence on this normalization.
Okay.
Okay, and then you do this dimension reduction. So this is also very similar to what you do for RNA data, and it's the same rationale behind it. You want to reduce noise. You want to. It's simpler for many of these methods to work with a sort of smaller representation with fewer dimensions, and also the first principal component often catches sequencing depth, even though you've normalized for it. The first PC or it correlates very well with sequencing depth. So you typically remove it when you then work with these components, for example, for clustering. And there's a lot. I mean, this is often like for single cell RNA-seq. There are many sort of parameters you can tweak, and you can play around with this. For example, which of these peaks you want to include, how many, if you want to choose the ones with highest signal or most variability in your data. This is an iterative process, right? You tweak these things until you come up with something that you like and makes sense. It's a bit subjective in the end. And then, this I guess you've seen. It's exactly the same as for RNA. When you have this reduced dimensionality, you can reduce it even more to get down to 2 dimensions, for example, with a UMAP, and you can do clustering, for example, with this neighborhood graph, like you do for RNA. And one thing that's a bit more tricky with the ATAC-seq compared to single cell RNA-seq is annotating these clusters, so finding out which cell type they represent. Because in RNA-seq, you have your marker genes, and you can just use them and you know what cell types you have. But here you just have these peaks. So what you often do is that you create some, it's called gene activity scores. So this is like a proxy for gene expression. So basically you just assume that if a gene has a lot of open chromatin around it, it's highly expressed. So what you do is that for each gene, you have a measure of how much open chromatin is around it, and that becomes its gene activity score, which often correlates quite well with expression. And then when you have that, you can do all these, you can just compare this to, because now you've gone from peaks to genes, and then you can do this to compare your data sets to all these different reference data sets and annotate your peaks. And yeah, you also want, you want to annotate your cells, sorry. Because what you also want to do is that you want to annotate your peaks. If you have a peak on chromosome 2 in position, 184,000, blah, 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 you want to see where that peak is in relation to genes, in relation to other things. So the easiest thing to do is, of course, to just annotate this to the nearest gene or transcription start site. This is not always correct, but it's a good starting guess, especially if this peak is very near to a transcription start site, then you can assume that this is in a promoter or something, or it has something to do with the expression of this gene. But if a peak is far away from any transcription start site or right between two genes, then you don't know what this is doing. And yeah, this is actually a bit of a problem. So people are trying, for example, to use this 3D data, like Hi-C, to see for each peak, which genes is it close to in 3D space when the chromosomes are, folded in the nucleus. If it's in the same TAD, for example, this topologically associated domain. But this is often quite a lot of work. So this is something that people don't really do routinely. Then another type of analysis that you can do with ATAC-seq that you can't really do with RNA-seq is that you can you're not only looking at genes, but you're actually, you can interrogate these peaks themselves and see, do they overlap with other features on the genome? Do they have specific sequence motifs? Things like that. You can query these peaks and see if there's an interesting correlation to genome sequences, transcription factor binding sites, other data sets, SNPs, whatever you're interested in. So this you can typically do you can, for example, look for peaks that are specific to a certain cell type or to a certain condition, or any set of peaks you're interested in. Yeah, differential DNA accessibility, but this ATAC-seq version of differential expression. So this is basically which regions change the DNA accessibility between two different sets of cells, between different cell types, for example. And yeah, it's a very similar problem. The statistics look a bit different since here you only have zeros and ones, basically. In single cell ATAC-seq, you have more different numbers. You can have, up to hundreds or maybe thousands of reads sometimes for a single gene. Here you just have zeros and ones. But otherwise, it's the same sort of problem. You can have simple tests where you just compare two conditions, or you can have covariates, or whatever you're interested in. Okay, so that's basically it about ATAC-seq. Here are some different tools. This is maybe not completely up to date, so there might be some new things I've missed. Cell Ranger, ATAC Cell Ranger, it's the program from 10x Genomics, which can do all of these steps that we talked about. Often it's used at least for the basic, mapping reads to genome, doing the peak calling, finding out which cell each read came from for the sort of initial steps. Then what I've been doing, and I guess most people, is exporting this into R. So CellRut, I guess you are very familiar with now, and there is an add-on to CellRut called ChIP-seq, which is an R package that enables you to work with not just genes, but chromosome regions in CellRut. Yeah. So here you can do all these different things, normalization, filtering cells, clustering, UMAPs, differential accessibility, integrating data from various studies for various assays, a lot of stuff. This is very useful. And there's a Python version called episcanpy. So this is a Python version of scanpy. I'm not super familiar with this, but yeah, it does all the basic stuff. Cromvar is a package that was developed by this Greenleaf lab who came up with ATAC-seq. And it can do a lot of these things too, clustering and visualization and differential accessibility. But what it's good at that these other packages are lacking is motif analysis, for example. So if you have some set of peaks that you're interested in, what are the enriched motifs in that? Those kinds of things. And then I just want to mention this Giggle, which is a command line tool for cross-referencing genomic regions. So for example, if you have, say, 100 peaks or 200 peaks that are very interesting, 200 genomic regions that change between your cell types, and you want to see what they overlap with in many, many different public data sets, then this is a simple command line to do that. So yeah, it has nothing to do with single cell data in particular, but it's just a practical tool in some of these studies. Okay, so now ChIP-seq. So this stands for chromatin immunoprecipitation followed by sequencing. So it's a different method than ATAC-seq. And instead of just measuring open chromatin, this measures the interactions between a protein that you're interested in and DNA. So basically, you have your protein that you want to study, and you want to see where on the chromosome it binds. And the idea is quite simple. You have an antibody against this protein. You use that to fish out the protein and the DNA that was bound to the protein, and then you sequence that. And this method has been around for a long time. And yeah, the analysis is a bit similar to ATAC-seq. You also get reads stacking up like peaks, and you do this peak finding, and you can analyze these peaks in different ways. Unfortunately, this doesn't work well with single cell. People have tried it, and people have given up, basically. So here are some studies where they sort of got it to work, but they got very, very few reads per cell. So this was sort of enough to find different cell types, but you couldn't really do much with it. So instead, people came up with alternatives, and yeah, these I think were the first papers, and the methods are called co-batch or cut and tag, and it's the same method, and I think it's this name cut and tag that has sort of stuck. Now this is just called cut and tag, and this is actually very similar to ATAC-seq. So the way this works is that you have this transposase, and so you have basically you have your protein you want to study, you have an antibody that binds to this protein, and then this antibody recruits a protein complex with this transposase, right? So it's like ATAC-seq, but it's not towards all open chromatin, but now it's targeted to just the places where a particular protein is binding to the genome. And you can, so in a way, the assay is very similar to ATAC-seq. It also works well for single cell, and you can do it with the 10x droplet method. You can do it with the combinatorial indexing. The analysis is also very similar to what you do for ATAC-seq. But now you have this, a particular protein you want to study, and you just get the regions where the protein binds. I should say that this is not as sensitive as ChIP-seq, but it works with very few cells or even single cells. And of course, the thing here is that for every protein you want to study, you need a different antibody. And so it sort of becomes its own experiment. Like for every antibody, you have to test that this antibody really works. And yeah, for some things that a lot of people study, like histones and histone modifications, there are well-tested antibodies that you know work. But if you have some very exotic transcription factor that nobody has ever looked at, then it's harder. Then you have to sort of come up with the antibody and validate it yourself, and it's a bit more work. And yeah, this is not as, I mean, this is much newer than single cell ATAC-seq, so it's not as established, but there are papers coming out. So this is from, yeah, people associated with Scilab Lab and KI. So here they did single cell cut and tag to measure different histone modifications. And what you can, yeah, the data analysis I don't have to repeat because it's almost exactly the same as you did for ATAC-seq. And right now, I don't think there are any sort of commercially available methods. I could be wrong about that. But this is, I mean, it's not, if you know how to do ATAC-seq, then this is not that hard to do. You can even use this, like you can, with some tweaks, you can use 10x to do this. Another thing which has been around for a long time, but it's much harder, is single cell DNA methylation. Right, so DNA methylation is where you have a methyl group which binds to DNA, typically to a cytosine next to a guanine, so at these CPG sites. And if you have this, often you have many of these together at what's called CPG islands. So this is a C and methylation and a G. And if you have a lot of these methylations nearby, they're called CPG islands, and it's associated with repression of gene expression. So this often, not always, but in most cases, reduces or turns off gene expression. And the classical gold standard method to measure that is with bisulfite sequencing. So here you treat your samples with bisulfite, and that for every C that's not methylated, it converts this to a uracil, which will then be read as a T. But for the methylated Cs, they stay Cs. So this is what you typically do in bulk. But then this is a quite rough treatment of the DNA. So you destroy a lot of the DNA. So if you just have DNA from a single cell, 2 copies, then you lose a lot. So there are other methods also with restriction enzymes, for example, that can only bind and cut to DNA that's been methylated. And yeah, other fluorescent-based methods. So yeah, this paper is a bit old, but already many years ago, here are all the different, this tree describes different methods for doing single cell DNA methylation studies. And you have the bisulfite sequencing based methods, and you have the bisulfite free methods, for example, use restriction enzymes. And this is, I mean, people have tried a lot of things. Nothing has really stuck. Nothing has really become super popular because this is still very difficult to do. It's difficult to analyze. It's a bit shaky. So, I mean, people have been doing this, but it's not as popular as ATAC-seq, for example. One problem is, for example, also that you get very uneven coverage of the genome. So maybe for some sites, in some cells, you get data on some CPG sites, and in other cells, you get data on other CPG sites. And overall, you lose many of these CPG sites. So this is something that's possible to do, and people are doing it. And I think even people at Scilab Lab are doing it, but it's not like a mainstream thing yet. Yes, Åsa?
I can just comment. I know that they developed one protocol for this at NGI in Uppsala together with like Jessica Nordlund group, and they offer single cell bisulfite sequencing as a service also.
They do, yeah.
But I mean, one big issue with this is the cost, because you amplify the genome, so it's basically doing a whole genome sequencing almost for every single cell.
Yeah.
It's quite expensive sequencing-wise to do these experiments. So I think that's the main reason, because if we could do 100,000 cells, then we could have statistics and do good analysis, but with 200 cells, it's not enough.
Yeah. Yeah, and if you want to be able to call like a CPG site methylated or not methylated, you need some reasonable coverage of that site, right?
Exactly.
If you just have one read, then it's a bit shaky. Okay, moving on. So then there's been a lot of methods combining different assays to measure many different things, modalities, like they're called, from the same cells. So the most common thing is if you do both single cell RNA-seq and ATAC-seq on the same nuclei. So, for example, 10x Genomics has a commercial method for that. And there are many other methods like snare-seq and others. And this is a quite useful thing because then you can look at in exactly the same cells, you can see which genes are expressed and what does the chromatin look like. And yeah, I will get into this a bit more. Then there are methods for example, single cell RNA-seq together with protein abundance for then a selected panel of proteins, for example, site-seq. You can do single cell RNA-seq and DNA methylation from the same cells. This last one called dogma-seq, you sequence single cell RNA, you do ATAC-seq on open chromatin, you get the abundance of some proteins, and you also get the DNA sequence clonality information for mitochondrial DNA. So they say that this covers the whole central dogma, so they call it dogma-seq. But yeah, this is also a bit old, but already here there were a lot of methods. People were just like going nuts with this. And I mean, these ATAC-seq plus RNA-seq, this is quite popular. People do it. And site-seq is also, yeah, something that I've seen that people actually use. These other ones, I think they are a bit rare. I mean, this is still developing all the time, so maybe these methods will become more popular too. Okay. And how am I doing on time? Yeah, just still some time left. Okay, so multi-omics
I have a
analysis.
question also, sorry.
Yes, please.
Yeah, thank you, Jacob. So there is also now these kits for transcriptomics and single cell whole genome amplification. So that's the latest from BioScribe and QIAGene.
Sorry, for what? Transcriptomics and
Single cell whole genome amplification sequencing. So
yeah,
you can sequence the whole genome plus the transcriptome in the same cell.
yeah. That's I haven't been keeping up with this, so I'm sure there are these cool things I've missed. But thank you. What I have worked with is this where you do single cell RNA-seq and ATAC-seq together from the same cells, and this is quite nice when it works. So the way I've been doing it, and that I think is suggested in these tutorials, is to first analyze each data type separately, and then you can do you can combine data. For example, when you cluster cells to identify cell types, you can do that with the RNA-seq and the ATAC-seq data together using a joint neighborhood graph. So this is an option in Seurat or SynYac. Or you can, for example, cluster the cells on RNA and then see if you can find more subclusters using ATAC-seq. But this is quite now, yeah, with the Seurat and SynYac, this is quite well supported, and it's quite easy to do these things. I think in the beginning it was sort of a bit uncomfortable and not super robust, but now I think that yeah, it's not so painful to do this actually. Okay, some last slides on spatial methods. I don't know how much you covered this in this course, Åsa. Have you been talking
Not
about it?
much at all. So,
Okay, so this will be very quick. So Probably some of you have heard about this, that you have these spatial methods where instead of just having data from each cell, you have a tissue section, and you get data from where in this tissue section each data point came from. For example, you can have RNA-seq, and this is an example of a part of a mouse brain. In each dot is a section where you measure gene expression, and you can see that some genes are highly expressed in one region and the other genes are highly expressed in some other regions. So there are two basic flavors of this. One is called spatial transcriptomics, at least it used to be called that. So this is based on sequencing, and the good thing is that you can capture all genes, everything you can sequence, you will pick up. But this is not really single cell resolution. So each spot here corresponds to many cells. And the way this works is that you have your tissue section, you put it on an array, and then your transcripts diffuse onto that array. And depending on where they land on the array, they get different tags. So by sequencing these tags, you know where in the section each molecule came from. And then you have these imaging-based methods where the readout is by microscopy. And then you have to select a subset of genes you're looking at, but instead you get much higher resolution. You can know exactly which cell each transcript came from because you see it in the microscope. And sometimes you can even get subcellular resolution. And I will mostly talk about this sequencing-based methods, the spatial transcriptomics. So this is also a product from 10x called Visium, and this is also a product from 10x, by the way. But this is spatial transcriptomics has also been adapted to ATAC-seq. What is this? Yeah. So the way you analyze this is very similar to single cell data, but instead of cells, you work with spots. So each little region here. And yeah, there is a lot of work where you do both spatial and single cell together on the same material to both get sort of single cell data, and then you can sort of, with some computational method, map where on this tissue these different cell types came from. And yeah, this has been adapted to ATAC-seq. So now each little box is a region in a tissue section, and for each little box you can measure how much open chromatin you have in different regions of the genome. And then you can do all the, clustering and coding cell types and stuff. And this is also a bit new and experimental, but it seems to work quite well. So then basically, you can call different, you can see that different regions in the tissue stand out, and you can sort of connect this to different tissue types and cell types. And they have also managed to adapt this to cut and tag. So then you can measure how a specific protein interacts with the chromatin separately for each little box, each little region in the tissue section. So here you have histone 3, lysine 27 trimethylation, and then each box here is a small region of the tissue, and then each box here corresponds to each dot here, and you can you can tell different regions apart. You can sort of annotate them to see which tissue types they represent. And then here's another histone modification, and the third one. So this is still not widely used, but it's very cool that this is possible and that you get useful data out of it. A lot of this has been developed also by people at KI. Okay, so summary, we've talked a lot about single cell, which is very well established. You have these commercial kits, it's easy to do. The software is also, it's very mature and very easy to use if you know R. And we talked a bit about ChIP-seq and cut and tag. that this is possible to do for single cell. The analysis is very similar to ATAC-seq. DNA methylation, we just discussed very briefly, and then these multi-omics and spatial methods, just so you know it exists, basically. Here are some resources. This is Jakub website, so this is the R package that helps you work with the ATAC-seq data and current tag. I'm going to plug this course. I forgot to update this slide, but yeah, I don't know when it's going to be given next time. It's quite useful material if you're interested in this. Even if you're not going to take the course, you can sort of look at the slides, look at the labs. And then, yeah, 10x Genomics, if you're interested in their method, it has a lot of detail of how all these different analysis steps work. Yeah, and with that, I'm done, and it's still some time left. Thank you.
Thank you for the presentation. I have a question regarding the peak calling of ATAC-seq. Like, is it like region on, region off, or it takes all the cells and then bins through the genome and looks like for these cells, there are reads here and then stacks it and then calls a peak?
Yes, so it just takes all the reads together. You don't care about which cell each read came from. And then you find the regions with many reads. And then once you found them, you go back to the data and for each cell count.
You mark it.
Do I see a read in this region? Do I see a read in that region?
Okay. And the other question is regarding this ChIP score for, like, is it different? Is there a different cutoff threshold for single cell than in bulk? Because in bulk, you're expected to have more than 20%. In single cell, since you have not so many reads coming from a single cell. So the number of peaks that you get, like, is the ChIP score higher? So the cutoff is also higher. And one should look into that.
It's a good question, and I don't know, actually. I don't know it right away. Sorry.
Thank you.
Thank you, Jakub, for the very interesting lecture. So I have a couple of questions. So the first one is concerned with the DNA methylation. So I didn't really get what you mean when you were telling about some limitations of the methylation-sensitive enzymes, because shouldn't that be the most non-challenging or the most easiest way to perform, at least for the single cell?
Yes, so that is it's absolutely it's the much gentler treatment than the bisulfite sequencing, but it's still hard to get good coverage across many sites, for example, even with those enzymes. And yeah, and also you need to sequence a lot when analyze many cells.
So far the bisulfite will be mostly adapted, or actually it should be the most commonly used, right? Instead of the enzymes.
Yes, I don't actually know. I know people have tried both. I don't think any of the methods are sort of very commonly used. Åsa, do you know more about this?
No, me neither. I haven't really seen that many like large-scale studies or people doing it. It's mainly a lot of methods papers where people are showing that it's possible to do. And not
so
really,
when I last sort of kept track of this, I know that the people at NGI in Uppsala, they were sort of offering this as a experimental collaboration thing to try it out. But you say that now they actually have something that
I think it
users.
might still be at the experimental stage.
Yeah.
And I don't really know how many customers they're having for that part. I haven't heard anything about it. So if anyone else here knows, please let us know.
So I have just a second question regarding the ATAC-seq for the Vesium instrument. So just for my clarification, so is the augmentation process happening in the tissues first, then it gets transferred to the slides, or how it is happening in technically?
Yes. So the segmentation happens before it's loaded into this fluidics thing.
Yes. Okay. Thank you.
For the 10x multiomics, you have from the same nuclei a cell
Yeah.
barcode that is incorporated into the tag mutated sequence plus the mRNA. So then when you do the analysis, when you do the QC, I mean, sometimes you have suspicions of a lot of doublets. And you just said that it's very simple in the ATAC-seq data to infer the doublets. So could we do QC in the ATAC-seq data and then interpolate it to the transcriptomic data?
Yes. You can do QC on both, and you can see how well they agree, and you can use both of them for your filtering.
Great, thanks.
But maybe an expensive way to detect doublets is to do a ATAC-seq of everything, just to filter your RNA.
Yeah.
But it would be cool if we had that data.
But I guess with some data sets, a lot of benchmarking could be done with the different predictors of doublets, right?
Yeah.
That we have seen through the lectures.
And of course, I guess you have talked about the methods to detect doublets for RNA-seq data, right? Doublet finder and these ones. And for ATAC-seq, there are some other methods too that I don't remember the details of, but there are specific methods that sort of make use of the ATAC-seq data in a better way that you can use.
About data sparsity, do you know how many regions or peaks do you get per cell? Like if you have, because we have been discussing that sometimes you might have this subtype, which is only 5 cells. And then even if you combine regions or cells, that's still very sparse, right?
Yes. So if you have, I mean, typically if you have something like ATAC-seq or something very common, like looking at histone modifications, then you get in the tens of thousands of peaks. You might get like 30,000, 40,000, 70,000 peaks. And I don't know how many of those you have in each cell, but I guess it can range from some of these peaks are present in almost all cells, and some are very, very rare if they're just there in a very specific cell type, or they're just very transient or whatever. So I guess it will be from almost 100% of the cells to just a very small fraction. I'm not sure if it's a good answer, but it's the best I can come up with now.
So what you combine between cells to avoid the sparsity is the common peaks, or what?
Yes. So if you combine all the data, you will find the common peaks, and can also find peaks that are cell type specific, but it will be harder to pick up those, especially if you have some very rare cell types. So then what you can do is that once you've identified your cell types, then you can go back and redo the peak calling on just those cells, and maybe you can find something more.
Yeah.
I think also there has been some work with doing these sort of like mini bulks of like grouping several ATAC cells into these mini bulks to be able to analyze them together, because it's too sparse to just have each cell as an individual.
Yeah. So when you do these comparisons, you have to do them between groups, and they have to be quite big groups. If you want to look at differences, you can't just compare like 5 cells to 5 cells. It's not going to be good.
I mean, I think a general thing we should say about ATAC-seq data, we were talking a bit about that we need enough cells to detect different clusters. We need enough cells to do differential expression for single cell RNA-seq. And whatever you're trying to do there, you have to probably have 5 times as many cells to do ATAC-seq analysis to have stable signals. That even makes it even more expensive to do ATAC-seq. You need more cells. Because it's super sparse.
I have nothing to add.
I had a question. So usually when you do bulk and you want to basically check where the peaks are found, you kind of compare to a baseline expression or baseline presence of reads. So I work with cut and run, for example, and we use IgG in order to set like a baseline. So my question is, how does one do it here? How does one do it here actually?
So I think here you just yeah, it's a good question. I know that for ChIP-seq, people have been using, for example, input DNA, but here I think that I'm not 100% sure, but I think that for the single cell, you just use the sort of regions around that you identify some low signal regions.
Yeah. I was thinking that as well, like maybe it could be the same sample, either global distribution and then averaged or something around.
Yeah, I don't remember the details of this, but so when I've been doing this, I've been using the 10x, the ATAC-seq Ranger, and they have their methods documented. They have a special algorithm documentation, and I don't remember the details, but you can look it up there. But I know that there's nothing, you don't run an additional experiment to get a baseline. You don't do any IgG or inputs.
Yeah, that's what I was wondering actually. Cool. Thank you very much. It was a very nice lecture.